Hello students, today I am with you with a new topic from the molecular basis of the heredity, that is DNA replication, one of important topic from CSE point of view. This chapter molecular basis of inheritance is very important. You have to understand basic concept of the genetics in this topic, then you have to solve a lot of questions for your medical entrance. So from starting genetic material, characteristics of genetic material, then your experiment to prove genetic material, then this replication, transcription, translation, human genome project, rice genome project, DNA fingerprinting, all are important topic. So I am taking this topic, DNA replications. So what does it mean, replication? DNA replication, it is process of polymerization of nucleotide base pairs. न्यूक्लियोटाइड बेस पेयर्स की पॉलीमराइजेशन की प्रोसेस को कहते हैं रिप्लिकेशन। होती कब है, साइट क्या है इसकी, साइट कहाँ होती है, कहाँ होनी चाहिए, न्यूक्लियस में, न्यूक्लियस। होती कब है, एस फेज में, सिंथेटिक फेज। सिंथेटिक फेज ऑफ सेल साइकिल। अब क्या दोनों में प्रोकैरियोट्स और यूकैरियोट में सिमिलर होती है, प्रोसेस बेसिक सेम है, बिकॉज न्यूक्लियोटाइड जो डीएनए में होंगे वो टोटली सेम है। तो कौन कौन से न्यूक्लियोटाइड है, न्यूक्लियोटाइड्स डीएनए के एडेनिन, गुआनिन, साइटोसिन, थायमिन, 4 nucleotide. These 4 nucleotide will join in a definite pattern according to the parental strand. So requirement, क्या क्या चीजें रिक्वायर्ड हैं डीएनए रिप्लिकेशन के लिए। सबसे पहले डीएनए पी, डीएनए पॉलीमरेज, डीएनए पी, डीएनए पॉलीमरेज, देन nucleotide, then other enzyme helicase, then gyrase or topoisomerase, topoisomerase, then DNA ligase, then kinase, then RNAH, then in eukaryotic another enzyme telomerase, telomerase. These are the basic requirement of DNA replication. So we have seen that it will takes place in the S phase of the cell cycle, as well as site is nucleus, and why it will take place? It will take place to make their next progeny or any other progeny in the next cycle. So let's keep look to the process, but before this I would like to explain some of the fact. In prokaryotes, about 1000 to 2000 base pair are added per second by DNAP, but they are 50 to 100 base pairs per second in eukaryote. ऐसा क्यों? Because prokaryotic genome is much simply bind. Packaging of prokaryotic genome is very simple as compared to eukaryotic genome. In eukaryotic, there are large number of protein which bind this genetic material into chromosome or chromatin. So because of this complexity, but how this complexity is being overcome? This E. coli in this pattern, E. coli complete S phase means replication completed in 38 minutes in E. coli. Sorry, E. coli within 38 minutes. This time period is similar about eukaryotes also, because there are large number of origin of replication, ORI, origin of replication, origin of replication is a place from where the replication will start. In prokaryotes, it will start from the single point, but in eukaryotes there are the large number of the ORI, hence replication will be very fast in eukaryotes also. Though the process is slow, but it starts at the multiple points. So it again same things. Now here I would like to explain you type of replications. Types. So type of replication. Firstly, replication are of the different types. They can be semi-conservative, this is in most of organisms semi-conservative type of replication will take place. But does it mean? It means that if these are the two strands, then after replication they will form one new strand and one parental strand. This is new strand. This is parental strand. So semi-conservative. Then conservative. In conservative, this is parental strand. After replication, there will be one parental strand and another new, totally new strand. Both new strand are new. So this is conservative. Mean parental strand will conserve. Parental strand will conserve. Mean this is your parental strand. Parental strand. This is semi-conserved strand. New strand. Newly formed strand. Mixed type of the conservation also, replication. In mixed type, there, this is your parental strand. It will make some strand which will like this way. In another it is like this way. So new strand will form which will have different polarity. And this pattern so this is mixed type, mean combination of both new and older strand. So this is one type of the replication. Beside it, we will see other types. So I have to rub this one, or I have to rub this one. I am rubbing this. Give a look at here. Types of the replication again. Type of replication. In this we have circular, it takes place in prokaryotes. Second, in circular, this is genetic material and it will replicate in this fashion. Two strands, circular. Then theta shape. In this, replication will start from the center and there will be two copy like this way. Two copy like this way. And third one is linear. Linear as we seen in the eukaryotes. Mean simple if I make this pattern the DNA, then linear. This is fork, and replication will take place. The new strand will form in this fashion. Mean after replication, there will be four strands like this way. This is linear type of the replication. So circular in bacteria, and this is in viruses, this is in eukaryotes. Eukaryotes, linear. Eukaryotes. This is viruses. This is prokaryotes. Prokaryotes. So now we come to the process of DNA replication. So let's give a look to the process of the DNA replication. Suppose this is our double strand in this pattern. Now for replication, this strand will open. For example, replication start at this point. At point where replication will start is called 30-mer sequence. 30-mer sequence. It will be rich in adenine and thymine because adenine and thymine will have double bond and they can open easily. So 13-mer sequence and 9-mer sequence. 13-mer sequence. So this strand will open like this way. Like this way. So this thing, if I will make it at here, it will be called replication bubble. This will be called replication bubble. Replication bubble. And if there are some protein, single strand protein, they will be remain attached on this strand. These are single strand proteins. They will go away when the replication will start at the particular point. This half of this bubble will called replication fork. This half, replication fork. So you just give a look. That says half of the strand in this pattern and the remaining strand will be at here. So this half portion, either this one or that one. So no problem at all. So this sequence will be what we have seen in the structure of the DNA. That DNA two strand run anti-parallel. It means there will be one 5' end, another will be 3' end. Similarly, there will be 3' end and there will be 5' end. Now come to the start of the replication. Whenever this replication will start, this single strand protein will go away. So as soon as this strand will open, single strand protein will go away. This protein from there, this protein will go away as soon as this strand will open. So single strand protein, and why this strand will open? Because there is an enzyme helicase. Helicase enzyme will open both of the side of this strand, helicase, like this way. So this is helicase. Helicase will open the strand. Then because of opening of this helicase, there will be strain, means the further strand will condense at this point. So this strand will be removed by another enzyme called topoisomerase or gyrase, DNA gyrase. So this is topoisomerase and helicase. Now this strand will start its formation. So DNA polymerase cannot bind without a primer. So a primer will made by primase enzyme. So primer at here. This primer is RNA primer. It is being made by primase enzyme, and on this RNA primer, DNA polymerase can bind and can further continue the process. So if this is DNA polymerase, it can bind at here. So DNAP. Now this DNAP have a special characteristics that it can polymerize only through 5 to 3 direction. So at here the newly formed strand, this will be its 5' and this will be 3. So DNA polymerase will start forming DNA change toward 3'. So 5 to 3. So this DNA polymerase can add the nucleotide in 5 to 3 direction only. So it will start polymerization. Now this condition is reversed. This strand is called continuous strand. Or leading strand. Continuous strand or leading strand. Now give a look to the another strand. Here the new chain will form from 3 to 5, but DNA polymerase cannot form new chain in 3 to 5 direction. It can form in 5 to 3. So for 3 to 5, there will be some small primer. For example, this strand open till this time. So as much as this strand will open, new primer will form, and this DNAP will bind at here, and after its binding, it will form a small sequence 5 to 3'. Again, this is 5' primer. Again 5 to 3'. Again from 5 to 3'. 5 to 3'. As much as this strand will open, the RNA primer will form and DNA polymerase will make small fragments called Okazaki fragment. Okazaki fragments. And this strand is called lagging strand. Lagging or discontinuous strand. Lagging or discontinuous strand. So this chain will be action of this enzyme helicase, topoisomerase will remove the stain. Because of the coiling of the stain, this stain will be being removed by the topoisomerase. RNA primase will add the primer, and DNA polymerase will bind on this primer, and it will start polymerization in the 5 to 3 direction. So this is condition. Now what will happen on this lagging strand? It will also complete after some time. So let me rub this much of diagram. So what will happen now? Now this RNA primer being removed, and by RNAH was an enzyme. I have written that in the requirement. So after removal of this, so the two enzyme, one is RNAH and second is DNA ligase. So DNA ligase is also called molecular glue. So DNA ligase will bind this chain, and this lagging strand chain will also become complete 5 to 3 dash chain. So after this replication, we will have 4 strand, 2 parental, 1, 2, and 2 new strand. So this is semi-conservative. These are newly formed cells, मतलब daughter strand, and this is parental strand. So this is semi-conservative mode. Now there is one protein called geminin. This protein will attach at the initiation site, mean origin of replication, initiation site, but it will prevent second phase of replication. Mean after completion of first phase of replication, second will not carried out at the same time. It will takes place after the maturity. So this geminin protein will prevent the second phase of the replication within the same genome. If it will be absent, it will lead to the polyploidy condition, as we see in the polytene chromosome. This is the case. So geminin protein. Otherwise, if there is some fault in replication, then there are two proteins, CHK1, ATM1 protein. They activate P53 factor. This P53 factor will cause apoptosis, mean programmed cell death. If there is some major problem in the replication, then this protein will activate, this factor P53, and they will kill that cell. Because we don't have seen any child with 2 head, 4 arms, 2 through 4 legs in the society. So such type of discrimination, if come in the replication, then they will be killed by this protein through the apoptosis. Here is another fact that after addition of 10 raised to power negative 6 base pair, some error can take place. Error mean this DNA polymerase is though very sensitive for the replication, but after addition of this much base pair 10 raised to power negative 6, some wrong base pair can be added by this enzyme. So it is not as much perfect. As much of the mistake can also be takes place by this. But if this mistake is more and the major gene will change, then this apoptosis can take place. Otherwise, there is no any problem in this replication. Now here is one fact that where replication takes place. Replication can take place to synthesize the genome, to make the 2 copy. One will pass into the daughter nuclei. Secondly, we can write replication कहाँ होती है. First, replication of genome, replication of genome first. Secondly, site specific recombination. Here it can also take place, site specific recombination. Third, in pathogen attack. For example, agrobacterium attack in plant cell, then it will make its more copy within that plant cell, so they can format there. Fourth, they can form in chloroplast, CP and MT, chloroplast and mitochondrial genome. Fifth, it can form in PCR, polymerase chain reaction, in DNA fingerprinting. So these are the sites and locations where this replication can take place. First is to replicate the total genome. It means it will have to make the two copies of the genome, and one will pass in the daughter cell while another will remain in the parents. Secondly, site specific recombination or any type of recombination prepare. So where recombination is they have some fault, then this site specific recombination or recombination repair, new copy can form. Thirdly, in pathogen attack, if some pathogen attack in some living organism, for example coronavirus attack in the body, it can make its own genome copy in our own body. Fourth, chloroplast and mitochondrial genome, obviously they are the semi-autonomous organelles. It means they have their own genetic material and it can replicate on the demand. And the last, PCR, when we get some sample of the DNA from some culprit and we have to make large number of copy of this sample, then polymerase chain reaction, large number of the replication can take place. Now I would like to explain here telomerase. I have written one enzyme, telomerase. So I would like to explain here this working of this telomerase. So just a minute, telomerase. But and when this telomerase will work, so we will look to this also. Here in this case, when we will get the two copy, one is this parental copy and another is a newly formed copy. In this newly formed copy, there is some sequence of the primer. This is RNA primer. So this RNA primer is not required in the DNA, so we have to remove this RNA primer. So this RNAH enzyme, RNAH will remove this primer. But now, how we can add this sequence? This sequence complementary to this second segment. When this is if 5-2-3, then this is 3-5-3. So here some sequence are missed. We cannot add this sequence because DNA polymerase cannot add without primer. If the DNA polymerase have to add this sequence, then how? There will be some primer, again same problem, primer then DNA sequence. DNA polymerase cannot act without primer. So here this enzyme telomerase will work. Telomerase and replication problem. It will synthesize some chain by reverse transcriptase activity, and by making a loop, now it will make this sequence complementary to this strand. This sequence will be complementary to this at here. Complementary. So telomerase enzyme reverse transcriptase type activity, and it form the loop. After some time, this loop will remove and this strand will complete. So in eukaryotic, this enzyme telomerase act to solve the end replication problem. End replication problem is to remove this RNA primer and to resynthesize DNA from the complementary strand. So DNA polymerase cannot complete that process. This process can be completed only by this telomerase enzyme. Now, in eukaryotic, I would like to explain some of the facts regarding the eukaryotic. In eukaryotes, there are separate DNA, alpha DNA, delta DNA. These DNA work differently. So this alpha DNA will start this chain, and for Okazaki fragment, if this is a DNA sequence, then for this Okazaki fragment, it is believed that here this is primer. After primer, DNA alpha, this DNA alpha will make a short chain, and further chain will be made by this DNA delta. But in case of this lagging strand, this DNA delta does not bind, and this small sequence is made by only this DNA alpha. But here at this point, DNA alpha then DNA delta. So this is polymerase switchover. Polymerase switch over. It does not take place in this lagging strand. Hence, there is a small fragment in the lagging strand in eukaryotic. But there are different reasons in different case. So, okay, I have done with this topic DNA replication, and if you have any doubt, please you just text me. I will solve your queries whenever I will get the time. But firstly, give a clear look to this DNA replication, and I will be with you with some new topic very soon. Till time, thanks.